Hello. Today, we would like to share with you about the cytological diagnosis of metastatic malignant melanoma by fine needle aspiration biopsy. Let's proceed to introduction. In this part, I will explain about the definition of melanoma, diagnosis, specimen collection and treatments. What is melanoma? Melanoma is a cancer of pigment cell which is melanin cell. It happens when melanin cell grow out of control. The pigment cell which is melanocyte, mostly in our skin, it also protect us against sun. Melanoma also can grow at many sites such as mucous membrane, nose, mouth, middle ear and eye. It also acts to invade normal tissue and spread to other parts of the body. Next part, for clinical diagnosis, there are 6 clinical diagnoses of melanoma. They are ultrasound, MRI scan, CT scan, pigmented lesion assay, PLA, last but not least, as mentioned in our title, fine needle aspiration and biopsy. Let us focus on biopsy. There are 2 types of biopsy. First, it is punch biopsy where punch tool were used to remove a mole and the layers of skin underneath it. Second, it is excisional biopsy. Scalpel is used to remove the mole. Then we will continue with lymph node biopsy. Firstly, fine needle aspiration were conducted to check if melanoma has spread from the skin to nearby lymph nodes. Next, sentinel lymph node biopsy, which blue dye will be injected around the tumor to identify the lymph nodes where melanomas is likely to spread first. Other than that, melanoma can be categorized 5 stage, which are stage 0 until stage 4. Last but not least, these are several examples of treatments for melanoma. They're surgical, immunotherapy, targeted therapy, radiation therapy and vaccine. Now, we look at clinical history. Clinical history is to identify early and then can treat early before become worse. The first one is primary melanoma. It is early stages of melanoma. We look at the changes in pigmented lesions using ABT criteria. A. Asymmetry. B. For the uneven borders. The color is dark black or have multiple colors. D is diameter that more than 6 mm and have evolving change in size, shape and color. There are 4 types of primary melanoma. The first one is superficial spreading melanoma. This type is the most common. It have diffused borders, brown, black colors, irregular edge and have elevated surface. Next, nodular melanoma has dome-shaped, sharp borders, extend vertically and has smooth, warty or crusty surface. Lentigo maligni usually have for elderly people. There are flat, irregular shaped, smooth surface and spread along the skin. The last one is acrolentigus melanoma, which is usually at hand or sole of foot. It become brownish marked at first, then it become more darker and wider. It also has irregular border. Next, the patient also can have swollen lymph nodes, which is difficulty to swallow, and the swollen lymph nodes is firm and painless. Excessive UV light and have many moles also the risk factor to become metastatic malignant melanoma. It is because exposed to excessive sun can damage DNA. If the patient has many moles, can use ugly duckling method to recognize different one mole other than moles. We can see in the red box that mole has more darker than other mole. The last one is family history or gene mutation. It can carry inherited mutation from the parents. If they have phenotypic traits such as red or blonde hair, fair skin and light-colored eyes, then they have more potential to get this cancer. Here are cytological features that shows there is melanin production. The presence of melanin pigment in cytological smears is a significant indicator of melanin production, particularly in the context of diagnosing malignant melanoma. The identification of melanin production in cytological smears is characterized by dusty or finely pigmented granular and evenly distributed in cytoplasm. It can appear as yellow-brown granules in Pap stain, in Figure B, and black with MGG stain, as in Figure C. But melanoma is heterogeneous. So, some of the cells might be pigmented while others are not. In FNAs, melanin production rarely found because of variability of melanoma. They can be amelanotic, in which there is no pigmented cell shown. Background of FNA samples can also contain hemorrhage and necrosis. It may obscure the visibility of melanin granules. There is a need to distinguish melanin from other pigments such as hemosiderin and lipofuscin, which can complicate the identification of melanin in FNAs. For melanin ingestion, it is where macrophages phagocytize melanin from necrotic or apoptotic melanoma cells and usually occurs in primary and metastatic melanoma cases. Melanin have been found in macrophages in 20% confirmed melanoma cases. In this picture, melanin in macrophages may have coarse or clumpy character. This finding can be helpful or vice versa, depend on site of target lesion. So, additional immunohistochemical staining and clinical correlation, very often necessary. Example, Pigments in macrophages from aspirates of lung usually represent carbon pigments from inhalation of coal dust or smoking.
This is normal melanocyte. These cells are dark in nature and dendritic in shape. All melanocytes migrate from a region known as neural crest. Each epidermal melanocyte dendrites are utilized to transfer pigment granules to neighboring epidermal cells. Nuclear feature and placement. There are a lot of nuclear features that we have learned for atypical cases such as nuclear enlargement, high NC ratio, hyperchromatic, coarse chromatin, and so on. But in this case, metastatic melanoma, I will highlight some of the important nuclear features that will help to diagnose. First is presence of multinucleation. Multinucleation has been frequently seen in several published series, and some publications have even reported that finding in 100% of cases. Secondly, eccentric nucleus. If they are solitary, nuclei are usually eccentrically located in melanoma cells, imparting a plasmacytoid appearance to them. What is plasmacytoid? The plasmacytoid appearance in metastatic melanoma refers to a specific morphological characteristic observed in certain variants of melanoma, particularly in cases where the tumor cells exhibit features resembling plasma cells. How do we know it is plasmacytoid or plasma cell? We can observe both morphologies based on cellular pleomorphism. As we can see, plasmacytoid has high pleomorphism because of variable size and shape of cells, while plasma cell has minimal pleomorphism because of uniform appearance. Osteoblasts are also contenders as mimics of melanoma, but their nuclei are almost beyond eccentric and appear as though they might jump out of the cells. These are further information about the differences between intranuclear cytoplasmic and nuclear pseudoinclusions. Based on their definition, intranuclear cytoplasmic inclusions are structures formed by the invagination of cytoplasm into the nucleus, containing cytoplasmic material and organelles, while nuclear pseudoinclusions are invaginations of the cytoplasm into the nucleus that appear as vacuoles but do not contain true cellular organelles. Next, based on the presence of cytoplasmic material, intranuclear cytoplasmic may contain degenerated organelles, proteins, and other cytoplasmic components, often indicative of cellular stress or pathological conditions, while nuclear pseudoinclusions consist of cytoplasmic material without a defined membrane separating them from the surrounding nuclear content and are more likely to be artifacts. Next, for intranuclear cytoplasmic, they often show positive staining for certain markers, such as CK19, which can help in their identification during immunohistochemical analysis. It may stain for cytoplasmic proteins, such as keratin or other cytoplasmic markers. But for nuclear pseudoinclusions, these structures typically do not exhibit specific staining patterns and may not react positively to immunohistochemical markers, and also does not stain for cytoplasmic markers.
In most melanoma cases, most findings consist of a mix of polygonal cells, epithelioid cells and spindle cells. First, the polygonal cell is dominant in melanoma cases. It is characterized by moderate to abundant granular or clear cytoplasm and also has distinct cytoplasmic borders. Next, the epithelioid cell is another cell that is mostly found in melanoma cases. Its cytoplasm is similar to a polygonal cell. However, its cytoplasmic border is unclear. Next, the spindle cell is also a cell that is found in melanoma cases, commonly from subcutaneous nodule origin. It is characterized by its elongated shape with tapering cytoplasmic ends. Next, giant tumor cells. Giant tumor cell is a large multinucleated cell with hyperchromatic nuclei and abundant cytoplasm. It also may contain mitotic figures, which is another indicator for melanoma. We also can see pleomorphism in melanoma cases. In this figure, some cells are larger than the others. Multiple cytoplasmic vacuoles also is another sign of melanoma. The size of the vacuoles are usually under 1 micrometer. These small cytoplasmic vacuoles are best seen in MGG stain. Presence of cytoplasmic vacuoles is a distinction characteristic between melanoma and lymphoma. This is the smearing pattern and background details of metastatic malignant melanoma. Look at these examples. All are different slides, but all of them are hypercellular, with presence of different shapes of cell in each smear. In the first smear, we can see that the cells are round to oval. The second is epithelioid melanoma cells and the third is spindled cells. The cells can also present in single or cluster. So, the smearing pattern for melanoma cases can be different in each cases, depending on what kind of primary melanoma the patient had. This is an example of smear taken from the lymph node, which is the common site for metastatic melanoma. In the background, presence of abundant small lymphocytes confirms that this site of smear taken is the lymph node. There are also presence of red blood cells in the background, that can be caused by needle trauma, where the needle punctured blood vessels. The overall smear has dirty background. This is most likely consists of necrotic debris or degenerated cells. This is the example of smear taken from the liver. Smearing pattern here is predominantly single cells, but there are also small cluster and occasional spindled cells present. In the background, necrotic bodies and inflammatory cells can be observed. Diagnosing melanoma could be very challenging and confusing, by just looking at morphology of the cells. A way to confirm melanoma cases is to do cell block and stain using immunohistochemistry stain, either S100 or HMB45. These are the examples of positive results of the immunohistochemistry stain. The cells affected will be stained brownish, which indicates a positive result.